Okay, next section of the nerves part of the syllabus, 4.1.2, I, J, and K. We have to describe with the aid of diagrams the structure of a cholinergic synapse. So another new word for you to deal with there. Outline the role of neurotransmitters in the transmission of action potentials, and outline the roles of synapses in the nervous system. So first of all, just to orientate yourself as well again, so here we have a sensory neuron. Remember that sensory neurons have the cell body branching out from the side of the axon rather than in the end of the nerve like in a motor neuron. So in this diagram, the direction of the impulse would be in this direction, so from the receptors in the skin along the axon, and then to these multiple synaptic endings along the dendrites, these branches at the end. Notice there are multiple dendrites and therefore multiple nerve endings. And one of these synapses that we're going to look at, a synapse is a space between the end of one nerve and the beginning of the next. So if you like, if I drew the branches and the dendrites on the branch of the next neuron, which is going to kind of join onto this sensory neuron down here, you can imagine that these ones would run up and come close to these synaptic endings of my sensory neuron, but they don't quite touch. So this gap in here, which I'll just circle in green, so one of these gaps, so between that synaptic ending there and the synaptic ending of the next neuron, that space in the middle is the synapse. So the electrical impulse has worked its way using action potentials, and the action potential moves itself all the way along the axon to the end of the sensory neuron, where it's then going to join onto what in this case may be a relay neuron. But the electrical impulse has got to jump that gap, and it can't do that using electrical messages. And so a synapse uses a chemical messenger to send that impulse across to the other side. So if you just orientate yourself now to this diagram on the left, and this is an animation that we're going to use to explain what's going on. This green and blue blob on the top left-hand corner represents the end of the synaptic ending of the sensory neuron. Then we have this space that's called the synaptic cleft, and then this pinky area in the bottom right-hand corner is then the membrane on the side of the relay neuron on this side. So we've got to jump this gap. So the electrical message can only make it this far, and then it hands the baton over, as it were, to some chemicals to then kickstart an electrical message again on the other side. Important just to mention a couple of things as well, that I'm just going to get rid of that to give myself some space. A synapse is the whole of this area. Bear with me 1 second. The whole of this diagram. So this includes, the synapse includes, I'm just going to write the word in here, synapse. So the synapse includes what we call the presynaptic knob. So that's this long arrow, just bear with me again. Okay, so this is the presynaptic knob on this side, pre meaning before, so it's before the synapse. And then similarly on the far side, this side is then our postsynaptic knob. Okay, so the synapse includes the presynaptic knob, the synaptic cleft, and the postsynaptic knob. It's not just the space, it's everything that's before and after it as well. So this represents one of the synaptic endings, and we're going to have a look at a few of the structures in here. This is the presynaptic membrane, which is going to be important in a minute, and you'll see why. So this is our presynaptic membrane. And similarly, but I won't write it down, on the far side we have the postsynaptic membrane as well. So the action potential is going to have is going to arrive from this direction, and it's going to arrive. That's not a very good arrow, is it? Let's change that to something that actually looks like an arrow. I'm just going to go for this one instead. So our action potential arrives from this end of the neuron and makes its way and reaches the end of the sensory neuron. When it reaches the end, it causes this to happen. So we're just going to start the animation. So it's not shown on here, the action potential arriving, but imagine it has. And the first thing you see is these calcium channels kick into play. So in the synaptic cleft, you have a number of calcium ions, which you'll see in a second. It's just labeling a couple of things over here, and I'll come to that in a second. So the calcium ions move in. So let's just go back over that again. So the calcium ions are invisible at the moment, but they'll appear. And the arrival of the action potential causes the calcium ions to flood in through the membrane. Let's just move that arrow out of the way. Okay. Actually, I'm just going to stretch that to there. Presynaptic knob. Okay. So I'm just going to list over here the stages of what's going on. So step number 1, arrival of the action potential causes an influx, is a good word to use, examiners like that one, of calcium, and these are 2+ positive ions, calcium ions into the presynaptic membrane, and into the presynaptic knob, in fact. Let's write it like that instead. Okay, now this is important, and this comes up in other parts of the biology we're going to study in year 13 as well. The influx of calcium causes the movement of these vesicles. So these are membrane-bound vesicles, little circles, little balls of cell membrane, and contained within them is a neurotransmitter. And as the name suggests, this is going to transmit the nervous message for us. So these vesicles, at the moment, they can't do the job they need to because they're stuck deeper within the presynaptic knob. But the arrival of the calcium ions causes them to migrate, and they move. I'll just pause it there. So you saw that one moving. I'll just run back over that again. So we see this vesicle moves and migrates and fuses with the presynaptic membrane. So step 2. Calcium ions cause the vesicles to migrate to and fuse with the presynaptic membrane. Now these, as I said before, these vesicles are full of the neurotransmitter. The neurotransmitter can be any one of a number of different substances. I'm just going to label them down here. So this is our neurotransmitters, and I'm just going to list a couple of them. So we have typical one that you will come across is something called acetylcholine, which is often abbreviated to ACH. So that's the chemical, that's the purple blobs inside the vesicle. And what else do we have? We also have serotonin is another one that you may have heard of. We also have dopamine, and number 4, just sneaking at the bottom of the page there. Just scroll down a little bit. It's adrenaline. Now, again, you will have heard of adrenaline. So if you just go back to the specification point, describe with the aid of diagrams the structure of a cholinergic synapse. So if you were asked about the structure, you'd be asked to mention the vesicles, the presynaptic membrane, the calcium channels, and the cholinergic synapse. What that means is that this synapse is using acetylcholine as its neurotransmitter. So if you see the word adrenergic instead, an adrenergic synapse, as you may guess, adrenergic synapse is one that uses adrenaline as the neurotransmitter rather than acetylcholine. So that's all that means. All the word cholinergic means is that it's releasing acetylcholine. So what's going to happen next is that we're going to have exocytosis, and you come across this in year 12 biology, and you should know that that means that this vesicle is going to kind of explode, if you like, and it's going to release its contents. It's going to rupture, and its contents are released into the synaptic cleft, like so. We're just labeling a couple of things on this side first. So there we go, into the synaptic cleft, and you can see that these molecules of acetylcholine are heading for the receptors on the postsynaptic membrane. So next step to label on here, so this is part 3, is that the acetylcholine molecules bind to the receptors. Now, similar to stuff you've come across in year 12 before, these receptors have a specific shape. So these are acetylcholine receptors, so they will only bind with acetylcholine molecules. They won't bind with serotonin, dopamine, or adrenaline. So they are what we call specific, and that's a really important word for you to. I'm just going to pop that at the top. I'm sure you know the spelling, but a really important word to use when you're talking about the receptors on this side. So they will only bind with their equivalent shape neurotransmitter. So in this case, we're talking about acetylcholine. So what happens next, and it's not shown on this animation, is that once the acetylcholine binds with the receptor on this side, that is enough to kickstart an action potential on this membrane and then throughout this nerve. So at the top we had that dotted arrow was the arrival of the action potential here. So then a new action potential begins on this side and spreads down the neuron just like it did on the other side. So similar to what you learnt before, sodiums rush in, potassiums rush out, etc., etc. And the arrival of that acetylcholine was what caused that to happen. Now, if this acetylcholine stays in here and remains in the receptor, then this will just keep on firing and keep on producing action potential, which isn't good. We want that to stop at some point because we want some control over our nervous system. So what we also have in this synaptic cleft is we have an enzyme, and I'm just going to write the name of the enzyme again up the top where I've got some space, and it's a long name. So let me just change the font size before I run out of room. And this enzyme is called acetylcholine esterase. Like most enzymes, it ends in the letters ASE. And so the job of this enzyme, what it does, and you'll see it on the animation in a second, is that it will release the acetylcholine molecules from their receptors and break them up into their individual parts. So an acetylcholine molecule, I'm just going to write this over here, running out of space a little bit. So acetylcholine breaks down into something called acetate and also a choline molecule. So that's the two parts that it's broken down into. So then it releases from the receptor and the action potential stops firing on this side. When the acetate and the choline are produced from the acetylcholine, then those molecules are released back into the synaptic cleft, and they will go back over to the presynaptic membrane. Just play the animation. And this is where the transporter protein kicks in, because it's going to take the acetate and choline and put them back into the presynaptic membrane. You can see some going in here. And it's going to reform them, so acetate and choline back together again. There comes the enzyme. Let me just repeat that so you can see the enzyme coming along. So it collects any spare acetylcholine and also the stuff from the receptor. And the acetate and choline molecules are bound back together again in here, in the presynaptic knob, to form acetylcholine, where new vesicles are formed, waiting to then to be reused next time an action potential arrives. That's pretty much it for what happens with synapses. In the class, I'm going to ask you about suggestions for why you think it's necessary to have synapses. Why do we not just have one great big long nerve? So think about, before you come into the lesson, why might it be that we have these spaces? Because it's only going to slow the process down, sending these chemicals across the gap. But there are a few very good reasons that we have these gaps and that no two nerves ever touch. A little smiley face to finish with. I hope you've understood that. Oh, he's a bit fat. Never mind. And we're done.